Efficient generation of transgenic mice by direct intra-ovarian injection of plasmid DNA.
Obtaining transgenic mice by injecting enhanced green fluorescent protein, EGFP for short, into the plasmid of the ovaries of a fertile mice. Now, they determine the frequency of the transgenic mice by pairmating, PCR, DNA sequencing, analysis from tails of offsprings. Now, the question in hand was if oocytes, which are immature female sex cells, could take up and transmit exogenous DNA by injecting the circular plasmid DNA in vivo by electroporation. Now, the objective was to evaluate a novel transgenic technology involving direct intra-ovarian injection of plasmid into mice. Now, their hypothesis was that foreign DNA enters the oocyte by penetrating the ovarian follicle and crossing the zona pellucida of the oocyte within the follicle. So in past research, Levitorno and his colleagues have shown that sperm cells can bind and internalize exogenous DNA, and they can subsequently transmit that DNA to their offsprings. Pronuclear microinjection was used, which is a widely used method of germline genetic transfer. Now, it is used because it is such a cheap cost, so it's very beneficial. Now, different methods that were used were retroviral infection, embryonic stem cells. Now, these are very costly, so they don't really use these. Now, the cool thing about this was the gene of interest was not only integrated into the genome, but it was also transcribed, translated into a functional protein, and transferred to the next generation. Who knew?
The animals that were used in this experiment were 5-week-old female Kunming mice purchased from Peking Union Medical College, housed in one room for more than 1 week, constant temperature of 20 to 23 degrees Celsius, with a constant humidity of 60-70%, with 12 hours of light and 12 hours of dark. Intra-ovarian injection of the EGFP, enhanced green fluorescent protein expression vector. 11 adult female mice had their ovaries surgically exposed in a solution containing PIRES, EGFP, and 0.1% trypan blue directly injected using a micropipette. Mock injections were given using phosphate buffered saline, PBS, plus 0.1% trypan blue. After suturing and 2 days of rehab, they were mated with male mice of the same line and maintained in the same conditions.
Enhanced green fluorescent protein observed in follicular cells and oocytes as dark brown deposits. About 24 hours after the direct ovarian injection, the plasmids were dispersed and were absorbed by various ovarian cell types. If the exogenous DNA is successfully integrated into the nuclei of oocytes, it should be expected to transmit to embryos during fertilization.
In situ hybridization, ISH, of paraffin-embedded sections. Injected ovaries were fixed in 4% paraformaldehyde and PBS at 4 degrees Celsius for 1 day, before being embedded in paraffin sectioned and subjected to ISH. 454 nucleotides of EGFP from PIRES EGFP were deoxygenin labeled for use as a probe by polymerase chain reaction with the following primers. Ovaries were counterstained for 5 to 10 minutes using nuclear fast red. Tissues were microscopically photographed under bright field illumination to detect the expression of EGFP in the F1 generation. ISH was performed with the gonads of some of the transgenic animals using the same procedure.
Before the in situ hybridization, Hemilum and eosin were used to stain several testicular and ovarian sections where hybridization signals were detected at all stages of germline cells. GFP was expressed in both ovaries and testes of adult transgenic mice.
Superovulation and embryo culture. 2 days after injection, mice were superovulated. Females were immediately placed with males for mating. After 1 day, females were examined for presence of vaginal plugs, but females were killed by cervical dislocation and the oviducts were removed. The ampullae of oviducts were opened using dissecting needles, and zygotes with their cumulus cells were placed in a drop of M2 medium and cultured at 37 degrees Celsius in a humidified atmosphere of 5% CO2 in air. Embryos were examined under fluorescence microscope for GFP expression.
The green fluorescent protein was detected in at least one blastomere for 23 embryos under the fluorescent lighting. Of those 23, 17 were non-mosaic since GFP was present in all of the blastomeres.
PCR analysis and sequencing in the F1 and F2 generations. The transgenic mice was identified by PCR using DNA from the tail. Screening in genomic DNA was performed. Coding and gene regulatory regions were screened using primers. All these reactions were performed in triplicate. Animals were only considered transgenic when the sequence of PCR amplified products was identical to that of the EGFP gene.
The production of transgenic mice began with the original 11 female mice. Of those 11, 4 died from anesthesia overdose. The remaining 7 treated mice each had 19 litters of F1 offspring. The 19 litters of mice had a total of 171 mice, and based on the amplification of a 454 base pair product, 111 were identified as transgenic.
Chromosome preparation and fluorescence in situ hybridization, FISH. FISH analysis was conducted on 3 to 4 week old transgenic mice who were injected intraperitoneally with 2.5% colchicine. The PIRES EGFP was digested using XOI and BAMHI. 1.1 kilobases of EGFP probe was labeled with DIG using the random priming reaction kit. The hybridization signal was detected using the fluorescent antibody enhancer set for DIG detection. Slides were mounted in an anti-fade solution containing propydium iodine and viewed under Leica filter set 13. In conclusion, these experimental methods provide evidence that support the hypothesis that oocytes are capable of efficiently taking up exogenous DNA. These methods can be applied to other experiments involving mice and gene transfer of other traits.
GFP locus array was located at different positions and on different chromosomes in the transgenic animals with a frequency of multiple site versus single site insertions at 85.71%. Overview. After the direct intraovarian injection procedure, all mice survived and produced 2 or 3 litters each. Although the first litters contained fewer mice than the second and third, no other negative effects were observed. Of the 171 F1 offspring, 111 were transgenic, or 64.9%, with the F2 progeny producing 66.94%. In conclusion, direct intraovarian injection is an inexpensive and simple mechanism to manipulate embryos and result in a high rate of transgenic mouse production by efficiently integrating the transgene into the genome and transmission to the progeny. Simply put, DII resulted in lots of glowing babies! Yay!
Implications. Intraovarian injection is a useful technique with a great success rate for producing transgenic mammals by the integration of the transgene into the genome of a stable progeny. The high success rate shows that the simplicity and cost-effective intraovarian injection is a great technique for gene targeting today. Questions for DII. The direct intraovarian injection, or DII, was displayed in the female mice's ovaries. Therefore, a question that could be asked pertaining to this exact research study would be if the same procedure could be done within the testes of a male mouse. And if so, would it have the same simplicity and success rate? An additional question that could be asked is if gene targeting and production of transgenic mammals by DII would be just as successful with more complicated and larger organisms, such as flamingos and elephants, or even humans would be possible. Even though human genetic targeting has been shown recently to have multiple religious and ethical contradictions,
The possibility of successfully doing so with the simplicity of DII shows a huge step in our knowledge of the human genome, displaying some of the possibilities for gene targeting in the future.